Hey, thanks for joining me. This is the Adobe Fikemba theory. And today we're going to be learning biology. Okay? Biology. So biology, we've been discussing cell structure, function, and its components. So for you to be able to understand the level I'm in now, the part I'm in now, I'd like you to try and watch the previous video of biology, part 1, part 2, part 3, part 4, as the case may be. Talked about the description of what cell. We started with that. So I'd like you, if you have not followed me, follow me so that you can get more videos of all the teachings in physics, chemistry, and biology. Now, so we're going to be looking at cell structure, okay? Cell structure. We're looking at cell structure using the light microscope. So, cell structure using the light microscope. I want you to take note of something. Now, for the cell to be studied using the light microscope, the plant or animal material is cut into a thin section. So for the material that is going to be studied, to know what their structure looks like, the plant or animal material is cut into number 1. Take notes. The points are what I'm going to be highlighting here. Into number 1, into a what? A thin section. Okay? This is to allow easy visibility and entrance to seeing, giving you a clear view about what that cell material looks like. So that thin section will allow that happen. So, and then when it is cut into thin section, number 2, it is then stained with a dye. You see it now? Stained with a dye. You see? So it is stained with a dye. Do you understand that? Number 3. Now, once it is stained with a dye, the reason is because these thin sections are made up of what? Chemical substances, these cell components. So take note of another point. These cell components, okay? These cell components in these thin sections are made up of what? Chemical substances. So these chemical substances can absorb this dye such that when it begins to absorb this dye, it will not make this component, the dye, to be unevenly distributed or unevenly stained. It will not make this cell itself, that's the component, to be what? Unevenly stained. So with this unevenly stained dye on the cell itself, what will now, with that in the light microscope, it will now be very easy with the dye on those structures to know what they look like in terms of shape or form. Are you getting it now? Now, if number 5, let me just put that down. If the dye used is of different colors, so if the dye uses of different colors, the implication, so different color dye. Let me just put that as a point for you to hold. Different color dye. So if the dye used is of different color, what will happen is that on the light microscope, you'll be seeing different colors on the various components of that particular animal material, that's as the cell. Do you understand what I'm talking about now? So it is from this description or from this analysis, number 6, that scientists were able to do what? To describe the cell structure. It is from this analysis that scientists were able to do what? To describe the what? Cell structure. When you talk about the cell structure, you're talking about the cell shape or the cell form. And from this analysis, it is discovered that the cell, number 7, I'm just numbering it so that it will be easy for you to understand the step by step. That's why I'm doing it, okay? It's not like it was numbered, but I'm just doing that for you. So, scientists were able to describe the cell structure using this light microscopic analysis. Now, that now makes the scientists understand that the cell is made up of a what? A living component. So, the cell is made up of a what? A living component. It is from this study using the light microscope. And this living component is called protoplasm. You see it now? Protoplasm. Can you see it now? So this living component is called the protoplasm. And this protoplasm is enclosed by a membrane. We call that membrane what? Plasma membrane. Can you see it now? This plasma membrane is found at the outer part of animal, but it is found in the innermost part of plant. So what you see in plants is what? The cell wall before you see the cell membrane. So you see the cell wall, okay? In between the cell wall and the plasma membrane, you have what we call the middle lamella. Okay, when we begin to describe the various components and their function, we will break them down and explain them one after the other. So take note, the living component is the protoplasm, enclosed by the what? Plasma membrane. This plasma membrane is the outer layer of the animal cell, while the cell wall is the outer layer enclosing the plasma membrane. I want you to also know that this protoplasm is made up of the what? Cytoplasm. So protoplasm is made up of what? Cytoplasm and the what? And the nucleus. I'd like you to take note of that. So the protoplasm made up of the what? Cytoplasm and the nucleus. It is with the help of the light microscope we are able to do what? To study and understand how the cell structure looks like in the light microscope. In the next class, we'll be looking at the cell structure and function, especially the cell function, using the what? The electron microscope. And if you look at that, you will see that there were some separation technique method that were applied. One of them is chromatography that was used. Another one again was centrifugation, the modified form of decantation. So you could see this is very wonderful. In the next video, we are going to discuss cell structure using the electron microscope. I hope you enjoyed the video. I'd like you to follow me if you have not followed me. Okay? Share the video. Thank you. See you in the next class, biology. Have a wonderful time.